Good morning. First of all, I would like to thank the organizer for giving me the opportunity to present our results in this meeting or in this session about novel biomarkers in neurodegenerative diseases. Our current knowledge about prevalent neurodegenerative diseases has drastically increased in the last 2 decades, and this has been mainly due to a successful combination between basic neuroscience research, clinical findings derived from multi-center studies, and the use of cutting-edge technologies to search for reliable biomarkers in preclinical population. One of the best examples of this fruitful collaboration has been our current knowledge about brain changes occurring in the years preceding the first clinical symptoms of Alzheimer's disease. And this knowledge has led us to a very detailed map of the natural course of Alzheimer's disease, putting more emphasis on asymptomatic and prodromal stages, and providing a framework to integrate how the cascade of neurodegeneration relates to morphological changes in neurons, synaptic dysfunctions that finally lead to macroscopic brain changes and functional deficits in connectivity typically observed in MCI and clinical stages of Alzheimer's disease. A novel framework has been recently proposed in the field of preclinical Alzheimer's disease. In this framework, neuropathological changes start to appear years preceding the first cognitive and behavioral deficits in asymptomatic subjects. In fact, this has challenged the use of imaging and biomarkers in liquid to detect AD pathology at very early stages of disease. And in this scenario, it has been suggested that PET amyloid imaging liquid biomarkers are more effective to detect changes in AD pathology in asymptomatic stages of disease, whereas structural imaging biomarkers like atrophies of medial temporal lobe or cortical thinnings are more appropriated to track disease progression in prodromal and clinical stages of Alzheimer's disease. Next, I will focus on how different brain and behavioral systems are affected in MCI subjects. First, I will show you some correlates, hippocampal correlates of episodic memory deficits in this population. Next, I will present some data supporting damage of structural cortical networks in MCI. I will also present results about structural changes in the basal forebrain of MCI subjects. And finally, I will present some recent evidence supporting the link between amyloid concentration, sleep deficits, and cortical thinning in amnestic MCI. Evidence from postmortem studies suggests that first AD lesions occur in or appear in the entorhinal cortex and the CA1 layer of hippocampus, that both regions are strongly involved in the formation and consolidation of episodic memories and, are strongly impaired in Alzheimer's disease. And we also know that these two regions, entorhinal and CA1, are the main exit gateway from the hippocampal formation of information, from the hippocampal formation to neocortical networks for memory consolidation. And we have recently found that these two regions are significantly atrophied in amnestic MCI subjects. So, to evaluate if there is a relationship between damage of these two structural regions and episodic memory deficits in amnestic MCI, we have been using a cognitive paradigm in the lab with young adults, healthy elderly subjects, and amnestic MCI. In this cognitive task, we first presented faces of famous people preceded by a biographical cue that was related or not with the face that we presented before. And the face was presented randomly presented in a different quadrant of the screen, and we asked participants to press a different button to indicate if the face was related, semantically related or not related with the biographical cue. So here we have two examples of trials. On the left, you have the face of Fernando Alonso preceded by Formula One. That would be a congruent trial. And on the right, you have the face of Antonio Banderas preceded by the name of Julio Iglesias. That would be an incongruent trial. So by using this cognitive paradigm, we were able to evaluate the effect of previous semantic knowledge on hippocampal-dependent episodic memory performance. So in 1 hour later, in the memory task, we present the same faces as before, that part of them were presented in the same quadrant of the screen or in a different location. And we asked the participants to press a button as quickly and accurate as possible to indicate if the face was presented in the same or a different location. So we first found that the semantic congruence, that's to say, the association between the face and the congruent biographical cue, improved associative memory in young adults and healthy elderly subjects. And this memory benefit was significantly correlated with the spectral power of theta oscillation in parahippocampal regions, supporting the role of hippocampal oscillation in episodic memory, at least in these two populations, in young and older adults. However, MCI subjects didn't obtain benefit from semantic congruence in the associative memory task. But when we divided the MCI subjects in carriers and non-carriers of the APOE4 genotype, we found that only those who were non-carrier of the APOE4 genotype obtained benefits from semantic congruence in the associative memory task. We also showed that the different relationship between episodic memory benefits and the integrity of the structural integrity in the entorhinal cortex and the hippocampal layers allowed us to differentiate between both groups, healthy elderly subjects and amnestic MCI, supporting the role of these two structures in episodic memory deficits in this population as well. Damage of structural cortical networks can be also detected in older adults at risk to develop Alzheimer's disease with sub-millimeter precision and using neuroimaging analysis techniques. Here we have the result of comparing pattern of cortical thickness between healthy elderly subjects and amnestic MCI, 100 participants in each group. And cortical thinning, as you can see, cortical thinning in amnestic MCI was limited to regions that are affected by early neuropathology in AD postmortem studies, like, for instance, the entorhinal cortex, parahippocampal regions, and temporal gyrus bilaterally. When we divided the MCI subjects into APOE4 carriers and non-carriers, we found that thinning of the cingulate cortex in different regions of the cingulate cortex allowed us to differentiate between the two groups. For instance, in the case of APOE4 carriers, we found that atrophic of the cortical, of the cingulate cortex was moved to the most posterior part of these regions and restricted to the right cortical hemisphere, whereas in the case of the APOE4 non-carriers, we found that atrophies were more anterior and limited to the left cortical hemisphere. These results suggest that a different pathway for neurodegeneration within the cingulate cortex might be underlying MCI subjects depending on this genotype. Evidence also suggests that the basal forebrain cholinergic system is affected in Alzheimer's disease. And this has been confirmed with immunohistochemistry experiments showing significant cholinergic denervation in cortical regions affected by AD pathology, and also with PET imaging revealing a significant decrease of acetylcholinesterase activity in AD regions affected in this type of population. However, there are few in vivo studies showing structural changes in the basal forebrain in amnestic MCI patients. So by using a probabilistic atlas of the basal forebrain, of the different basal forebrain cholinergic compartments obtained from postmortem human brain, we were able to compare volume changes in each cholinergic compartment of between healthy elderly subjects and amnestic MCI. I'm going to explain a little bit in this figure. In blue, you have the entire basal forebrain, and the yellow and orange spots reveal the significant changes, volume changes, in amnestic MCI patients when compared with healthy elderly subjects. All these changes were found in the CH4 nucleus that corresponds to the nucleus basalis of Meynert, where are most of the cholinergic neurons innervating the neocortex in the human brain. These changes in the nucleus basalis were significantly correlated with other cortical regions, atrophies of other cortical regions that are affected in AD process, like for instance the temporal lobe, the superior parietal cortex, or the prefrontal cortex. And also we found that volume changes in the nucleus basalis account for cognitive impairment only in amnestic MCI, not in control. So these results together suggest that the basal forebrain cholinergic system is also affected in amnestic MCI subjects, and this might be used as an in vivo marker of disease progression in this population. Sleep disorders are one of the main troubling symptoms in Alzheimer's disease. And in fact, different studies have identified a link between amyloid concentration and sleep deficits, but in animals. For instance, Kang and collaborators showed that an increased amyloid load in regions affected by Alzheimer's disease was followed by a chronic sleep restriction, but more interestingly, the same research group found that sleep deficits appear just after the first amyloid deposition occurred in the brain of APP-PS1 mice. So these animals showed more wake, less REM sleep, and less slow wave sleep with amyloid deposition. So in general, these studies are suggesting that there is a relationship between amyloid deposition and sleep deficits, and this relationship might emerge at very early stages of disease. So to test this hypothesis in humans, and more specifically in amnestic MCI, we decided to record physiological sleep and to measure alpha-beta levels in plasma in MCI population. And we found that MCI subjects showed a significantly decreased REM sleep together with more fragmented slow wave sleep, and also increased alpha-beta levels in alpha-beta 40 and 42. And we also found a significant association between the amount of slow wave sleep fragmentation and the increased levels of alpha-beta 42, but only in amnestic MCI subjects. We also wanted to investigate if these sleep deficits are correlated with the cortical integrity in MCI subjects, and we found that REM deficits were significantly correlated with the thinning of postcentral gyrus and the cortical atrophies in the posterior cingulate cortex and the superior parietal cortex. The latter two regions are highly vulnerable to amyloid deposition in different stages of disease. So in general, these results are suggesting that we might extend the link between amyloid deposition or concentration and sleep deficit to humans, to early stages of disease, and also we provide evidence to link this result to the cortical integrity of this population. So thank you very much for your attention.
Thank you for the nice talk. We have time for one or two questions.
A very nice talk. Just when you were doing the basal forebrain volumetric analysis for the cholinergic areas, I mean, obviously the area that you chose overlaps with quite a lot of other structures, such as the nucleus accumbens and the dorsal amygdala. How did you make sure that the volume changes were actually specific to the CH4 and not any of those areas?
We used for this study a probabilistic atlas of the of different nucleus of the basal forebrain, and we decide on a threshold to decide the significance in each nucleus. For that reason was a probabilistic atlas. We decided that 80% of postmortem human brains have to be the CH4 in our changes obtained in our study. So 80% of the postmortem samples were matched our result in our study.
Have you analyzed the hypothalamus in the study where you were looking at the sleep disorders, because the hypothalamus is the main site for sleep regulation?
No, we didn't do that analysis in this study.
I have one question. So it seems that the sleep disorders in Alzheimer's disease, sleep is deteriorated quite early, like during the preclinical phase of Alzheimer's disease. So how important do you think this will be for clinical detection of Alzheimer's disease?
Okay. That would be an early marker, but together with other markers of disease, because you know in aging, sleep disorders is also affected, so you have to discriminate very well between early neurodegeneration and normal aging before taking this conclusion. So sleep by itself can be used for anything, but in combination with other markers of disease, of Alzheimer's disease, might be more very useful.
Okay. Well, thank you. Thank you very much. We will move to the next.